Now, Juraj Adamik, research scientist in the Parker Institute for Cancer Immunotherapy, will present us the molecular and immunologic profiling of CD8 T-cell responses in patients receiving a multiple antigen-engineered DC vaccine.
Good day, everyone. It's a pleasure to speak at this scientific symposium. I would like to thank the organizers for the introduction and an invitation to present our work at this symposium. So this work is coming from a dendritic cell cancer vaccine clinical trial in melanoma patients. So at the beginning of my talk, I'd like to give a brief background about melanoma and introduce the concept of dendritic cell cancer vaccines. And then I will introduce the clinical trial itself that will then transition into the data presentation. And I have no disclosures. So melanoma is the deadliest form of skin cancer, and it's highly metastatic. Now, unfortunately, the rates of the diagnosis have increased dramatically over the past decades. And because it's highly metastatic, it is often diagnosed in late stages. About 100,000 Americans are expected to be diagnosed in 2020. Now, despite the advances in the current treatment of melanoma and recent developments of immunotherapies, there's still a significant portion of patients that does not respond to these treatments or acquire resistance during the course of the treatment. So therefore, this is a critical health problem in the United States and world. So dendritic cells are the true connectors between the innate and adaptive immune system. The idea of dendritic cell, and I'm going to use DC-based cancer vaccines, is to basically induce new or expand a pre-existing functional and durable tumor-specific T-cell responses. So we want these dendritic cells to educate the adaptive branch, and specifically CD8 T cells, to attack and kill the tumor cells. So there is a diagram here of a patient example where blood is being leukapheresed and elutriated, where the dendritic cell precursors are being generated and matured. At some point, a tumor antigen loading process is happening where the vaccine product is prepared and infused back into the patient with hopes of sharing this tumor antigen presentation knowledge with the T cells. So in our case, we engineered adenovirally transduced vaccines with 3 shared melanoma antigens: pyrosinase, MART1, and MAGE6. So vaccines are usually safe, but the question is, are they effective? There's about 200 dendritic cell clinical trials or more today, but the efficacy of the dendritic cell vaccines is still questionable. In terms of our clinical trial, here's a brief summary of it, a schematic diagram, where here at the baseline, day 0, prior to that, a first set of leukapheresis has been conducted, where the vaccine product have been generated. That is then being infused 3 times into the patients, and at day 43, second set of leukapheresis is generated. Then, actually, patients are being divided into 2 branches, with or without high-dose interferon alpha treatment. And day 89 then concluding over the trial has another leukapheresis. So you can see we have 3 different time points for analyses. And there has been a wealth of molecular and clinical data coming out of this trial. We've used numerous things like cytokine serum profiling, molecular profiling of the circulating lymphocytes, and a lot of immunomonitoring of the lymphocytes of these patients, including tumor biopsies. So going on here, just to show you a quick overview of the responses in the trial, where we see some partial responders and, of course, some early progressors, unfortunately. And then we have also some no evidence of disease patients that were actually separated into good and bad outcome categories based on their time of progression. So good and bad outcomes, those are the 2 categories I will refer to. So the study objective of our trial, or actually the study work, was to analyze the cancer patient-specific T-cell responses. So I'm going to go ahead and to show you that, to confirm that the dendritic cell vaccines induced and activated a successful tumor-specific responses, we conducted ELISpot assays from total PBMC cultures. And these demonstrated that majority of patients showed actually increased in interferon gamma producing T cells in response to the antigens. And that indicated successful vaccination. Now, the closer look at the baseline responses shown at the bottom there showed that about half of the patient showed pre-existing functional T cell responses to the melanoma antigens prior to vaccination. But there were no associations between having these pre-existing responses and T cell responses and some sort of favorable outcome. Next, because 16 patients were HLA-G positive, we were able to use dextramers to measure the T cell frequencies and identify peptide-specific CD8 T cell responses. So on the left-hand side here, you can see changes in the CD8 frequencies at multiple time points, starting from baseline, continuing post-vaccination. And these are further broken down here in the middle diagrams by patient response. And the take-home message here is that we did not see any consistent trend in patient CD8 T cell tumor antigen reactivity among these groups after vaccination. And as with the trend observed with the ELISpot data, the majority of 16 patients here had dextramer positive cells at baseline. But again, these did not set a stage for a better clinical outcomes for the patients. So collectively, we saw that the responses to individual antigens did not significantly impact clinical outcome. But what we did, we actually took all the individual responses and to all three shared antigens and grouped them into one combined response. And what we were able to see here, based on these Kaplan-Meier plots, is that a vaccine-induced CD8 T cell response showed significant association with overall progression-free survival, with overall survival and progression-free survival. So going on, we then were interested a little bit more into effects of previous checkpoint blockade for these patients. So because many of these patients were in late stage disease, they often had previous therapies. And here we subdivided the patients based on receiving either anti-PD-1 or CTLA-4. And what we saw here was that TCL responses overall were higher in patients that had previously received one of the checkpoint blockades therapies. And here I'm first showing dextramer frequencies that show a small but significant difference based on the previous therapy for MAGE-A6 specifically. And going on, here I'm showing a frequency, dextramer frequency responses. And what we see here is that the only MA-specific T cell response that showed some sort of clear increase over time was, again, MAGE-A6. So that was quite an interesting result. And it suggests that the previous blockade of CTLA-4 or PD-1 can slightly amplify some of the MA-specific responses, and this difference can persist through the course of vaccination. All right, going on next, we were also interested in profiling the exhaustion molecules on these cells. And here I'm going to only share a PD-1 expression levels. And what was interesting was to see that the best responders, the two patients that were PR patients, actually showed really extremely low levels of PD-1 on their MA-specific CD8 populations. The other exhaustion markers did not correlate significantly with any kind of clinical response. So next we use nanostring profiling to look at the levels and really to deeply profile these circulating lymphocytes from these patients. And here are shown data for the protein counting data for these checkpoint molecules, PD-1 and CTLA-4. Now, what we saw is that in general, both of these molecules were elevated their expression levels were elevated in the bad outcome patients at both measurements, at the baseline at day 43. So we also analyzed this data by looking at time-dependent change. And even though these data didn't come statistically significant, they were certainly, at least for the CTLA-4 data, we saw a very close significance in showing that the CTLA-4 or PD-1 expression levels were higher in the bad outcome patients. We've also used univariate Cox regression analysis to kind of determine whether or not their levels at baseline or post-vaccine correlated with overall or progression-free survival, and indeed many of these did. So we've used pan-cancer immune profiling panel and CAR-T characterization panel in order to better understand the pathways and the gene expression profiles of these basically patient circulating lymphocytes. And the way we've done this comparison was based on outcome groups. So we separated the good and bad outcome groups at both baseline and post-vaccine treatments. And we saw certainly an interesting signatures or different gene expression changes. And when we looked at the pathway analyses, what we saw was the following. And again, just to position you, we have the red and blue denoted pathways that are either upregulated or downregulated based on clinical outcome and broken down at baseline or day 43. So what we saw is that a lot of interesting pathways associated with mTOR signaling, T-cell receptor signaling, B-cell receptor signaling were upregulated in the good outcome category, while strong type 1 interferon response signatures were associated with the worst clinical prognosis. Here is also shown that CTLA-4 pathway actually came up as upregulated in the bad outcome patients. Now, we've also correlated pathways with overall and progression-free survival. And what was interesting was that we saw the T-cell responses were strongly correlative with overall survival and B-cell responses with progression-free survival. And we also saw that the interferon type 1 signatures were present in the low unfavorable survival category. Now, lastly, I'm going to share some data from the tumor biopsies that we are actually currently investigating right now, where we actually applied several nanostring sequencing panels to. And what we actually saw is, and I understand this is a little bit of a complicated slide, and I just want you to focus your attention on these different signatures. And what we actually saw is some interesting hypoxia like angiogenesis and VEGF associated signatures that actually were correlative of bad clinical outcome. There are additional pathways like integrin, ECM, stroma-like components, toll-like receptor signaling, TGF-beta. Again, some consistent themes along the tumor microenvironment. Now, what was interesting is that the VEGF interested us because of some additional data that we have collected, including blood serum cytokines and whatnot. And we've actually looked at the pathway specifically related to these in both three panels. And what we saw, and it was really nice to see, that there were several genes that were consistently upregulated in the bad outcome patients across the different panels. So it was sort of a external validation of the data. So this is just kind of an example of the pathways signatures that we see in the bad outcome patients in the tumors. So collectively, I would like to conclude the talk by summarizing the data, where we show that a majority of our 35 patients were successfully vaccinated in this clinical trial, and not individual antigens, but the combined total of tumor antigen-specific CD8 responses were associated with favorable overall progression and overall survival. So that argues for the importance of T-cell responses induced by the vaccines. And the previous checkpoint therapy enhanced some of the tumor-associated antigen T-cell specific responses, and in some cases they continued elevated over time. And the two patients with the best clinical outcomes certainly showed a very low PD-1 expression surface markers. Analysis of PD-1 and CTLA-4 protein expression using protein counting nanostring panel in circulating lymphocytes showed increase in expression of these checkpoint molecules in the bad outcome patients. And what we saw is that a lot of high immune checkpoint type 1 interferon responses, T-cell exhaustion-like gene expression networks were associated with inferior clinical outcome in patients. And lastly, we are looking into the tumors and saw some interesting angiogenic-like extracellular or matrix stroma-like signatures that associated with inferior clinical outcome in patients. So with that, I'd like to thank my mentor, Dr. Butterfield, and some of the people that collectively helped with the work, Patty Santos, Dina and Paul, our collaborators, Tim Hobbs, also from the Parker Institute. He's in the bioinformatics department. He was instrumental in helping with some of the bioinformatic analyses, and also Sarah Warren from nanostring, from helping conducting this nanostring sequencing research. And this is a picture showing the Parker Institute for Cancer Immunotherapy. That is where we are located. And I'll be happy to take any questions.
Have you looked whether blockade of PD-1 during antigen presentation by dendritic cells had any functional impact on the tumor antigen-specific T cells?
So yes, we had actually engineered dendritic cells that were expressing anti-PD-1 antibody, and they were co-cultured together with the patient T cells, and we did not see any effect of blockade of PD-1 during the tumor antigen cross-presentation. So we did not see benefit adding that during the antigen presentation. However, we certainly suggest that there may be additional mechanisms, or perhaps the blockade of the checkpoints after the vaccination may potentially increase the benefit of the vaccine.
Did you measure the antigen expression levels in dendritic cells? And if so, did you see any correlations between antigen expression levels and T cell or clinical responses?
So yes, so we've looked at the levels of antigen expression levels, both in the vaccines, both by RNA and protein, and we did not see any correlations between the amount of expression of particular antigen in the dendritic cells and better T cell responses. So that was not apparent.
Did you observe any correlations between CD4 and CD8 T cell responses?
So we looked at, and actually a reviewer asked us that for our manuscript, whether or not there were any correlation between CD4 responses. So we have extensively analyzed CD4 T cells, just like the CD8 T cells, but we did not see a specific correlation in any way with the CD8 cells to any of the tumor antigens. So while we believe they're important, and many studies are coming out of their importance in immunotherapy treatments, we did not see a correlation between CD8 and CD4 T cell responses to these shared tumor antigens.
Did you analyze any soluble serum checkpoint molecules?
So yes, we have analyzed actually a whole panel of soluble checkpoint molecules, including PD-1, CTLA-4, but many of those actually did not come up in terms of a good signal. But what we saw was soluble expression of PDL2, as well as LAG3 and TIM3 showed some interesting correlations with clinical outcome. And I'm going to point out soluble PDL2 that showed a significant association done by hazard ratio analyses, as well as correlative analyses, that the elevated levels of PDL2 actually predicted a better outcome in patients. Now, while we don't know what it really means, there are some studies indicating that PDL2 may have some different and perhaps even activation-specific roles, but we don't understand what it means yet, but the clinical correlations were very significant for PDL2. So I'd like to summarize the study by saying that while most of the patients were significantly vaccinated in our clinical trial, individual tumor antigens were not predictive of clinical response. The combined total was certainly impactful in terms of clinical outcome, such as overall survival and progression-free survival responses. Nanostring signature profiling certainly demonstrated an interesting patterns in checkpoint molecule expression, such as PD-1, CTLA-4, and we show strong associations with, or their increase showed strong associations with inferior survival in patients. I would like to thank everybody for their attention. I'd like to thank again the organizers for putting this event together, and NanoString for sponsoring the event, and thank you.